Assalamu alaikum. Our topic is an application for molecular modeling, which is molecular docking. That is our content, and we will start with what is molecular docking. Docking is a structure-based technique which attempts to find the best match between two molecules. The aim is to achieve and optimize the conformation for both receptor and ligand and the relative orientation between protein and ligand such that the free energy of the overall system is minimized. Docking can be between protein-ligand, protein-protein, and also protein-nucleotide. Steps of docking. Step 1. Start with crystal coordinates with target receptor. For example, here HIV-1 protease is the target receptor and the aspartyl groups are its active site. Step 2. Generate molecular surface for receptors. Step 3. Generate spheres to fill the active site of the receptor. The spheres become potential locations for ligand atoms. Step 4. Sphere centers are then matched with the ligand atoms to determine possible orientation for the ligand. And finally, Step 5. Find the top scoring or the best ranking. There are 3 scoring schemes, which are shape scoring, electrostatic scoring, and force field scoring. There are several applications on molecular docking. As virtual screening, docking with a scoring function can be used to quickly screen large database of potential drugs in silico to identify molecules that are likely to point to protein target of interest. Other example is lead optimization. Docking can be used to predict in where and in which relative orientation a ligand point to protein. This information may in turn be used to design more potent and selective analog. And the mainly we talk about is bioremediation. Protein ligand docking can also be used to predict pollutants that can be degraded by enzymes. Our example is modeling based structural insights into biodegradation of the herbicide diren from lactase 1 from cerebrobiosis. So the idea is the herbicide diren is used in many agricultural crops and non-crop areas worldwide, leading to the pollution of the aquatic environment by soil leaching. Therefore, it is of interest to explore the potential use of cerebrobiosis lactase 1 in the biotransformation of this herbicide by using its enzyme lactase. However, the structure of lactase from cerebrobiosis is still unknown, hence a model of lactase was constructed using homology modeling. The model was further used to drug beta-methyl benzoate in the presence of 4 copper ions to analyze molecular basis of its binding and interaction. The ligand-protein interaction is stereochemically favorable in nature, and to establish the optimal docking simulation protocol, the redocking of the ligand beta-methyl benzoate on the protein-ligand complex of the template 2-HRG was performed because it is assumed that the crystallized final structure of the complex represents the lowest energy conformation and therefore the most stable one. In this procedure, the ligand was taken from its original structure and reduced by means of the software Autodock in order to find the conditions that could reproduce the pose of the model or crystallographic ligand with the root mean square deviation lower than 2 angstrom as well as 100% reproducibility. So the docking protocol was established by keeping the single bond of the ligand free rotatable and the enzyme treated as a rigid body and applying the Lamarckian genetic algorithm for search based at the already known active site location in the template structure. This protocol was then applied for the redocking of beta-methyl benzoate into the minimized structure of crystal-1 and in the docking of the compound diren obtained from zinc database in the template and minimized cercil-1 structure. The advantage of the docking in this example is to facilitate the location of the binding site and the binder itself, also to give the proper guidance in the selection of the best pose of an unknown ligand. The conclusion is that the structure model of the lactase 1 from cerebrobiosis is solved in this study has similar fold with lactase from Trametes trogi and the Trametes species. This work gives evidence suggesting that cerebrobiosis, a fungus commonly used in the bioremediation and degradation of lignocellulosic biomass, may also be used for the biotransformation of the herbicide diren, since there are no reports in the literature regarding this purpose. That is our references, and thank you.